The overall goal of this procedure is to culture human mesenchymal stem cells on porous bone matrix. This is accomplished by first isolating mesenchymal stem cells from human amniotic membrane, or AM-HMSCs. Next, the porous bone matrix discs are prepared to favor the correct distribution of nutrients for cells to grow. Then, the AM-HMSCs are cultured on the porous bone matrix discs. Finally, colony-forming units are counted, and morphology, cell proliferation, and cell adhesion are all analyzed. Ultimately, cellular morphology, number of colony-forming units, cell proliferation, and cell adhesion are analyzed through optic microscopy, staining with vital dyes, and scanning electron microscopy, respectively.
The implications of this technique extend toward therapy of bone injuries because it's possible to deliver the mesenchymal stem cells into macroporous biomaterials that mimic the natural bone.
To isolate mesenchymal stem cells from human amniotic membrane, hold the umbilical cord with one hand, and with the other hand, detach the amniotic membrane, which looks like a translucent sheet. If the chorion is present in the sample, manually dissect it to separate it from the amnion tissue. Use 5 milliliters of PBS to wash the sample 3 times to remove residual blood, and use simple forceps to remove blood clots.